Hi class, in this recording we are going to focus on gametogenesis and the female cycle. The two gametogenesis cycles will focus on spermatogenesis, the production of sperm in the male reproductive system, and oogenesis, the production of oocytes within the female reproductive system. And then we'll also talk about the menstrual cycle and how to recognize the different phases of the menstrual cycle. So let's dive into it. Here I have just some histology of the testes. I want you to be able to recognize what the testes look like for histology. And if I just look here, we can see these tubes or circular structures. Some of these structures, I'll go ahead and annotate here. Let's go with green. Here we have a transverse slice, so it looks kind of circular. And then here we have a longitudinal slice, so it's an elongated slice. And when we focus on these structures, I want to emphasize the seminiferous tubules, these things that I've highlighted in green, are within the testes, giving us this spongy appearance. And if I zoom in here, let's see how the internet cooperates with me, we can see masses of cells in between the seminiferous tubules. Here's a mass of cells right here. This mass of cells in between the seminiferous tubules are called Leydig cells. Traditionally, the new school term is the interstitial endocrine cells. These are the cells that produce the testosterone, which we associate with the testes. Go ahead and close that tab. And I have a cross-sectional slice of a seminiferous tubule, just a beautiful one highlighted here. So we can talk about the stages. Let me close. I'll leave that open. So we can talk about the stages of spermatogenesis, and we'll start from the outside in. So first we have our spermatogonia. Our spermatogonia are going to represent the thin, flat cells on the outer edge. Oh, too many links. There we go. So right here we have spermatogonia on this outer edge of the seminiferous tubules. Those are the not sperm cells, the stem cells that we use to ultimately make spermatocytes or sperm in our bodies. The next cell moving in is the primary spermatocyte. And when we're looking for a primary spermatocyte, it's going to be right next to the spermatogonia. So we have a primary spermatocyte, for instance, right here next to the spermatogonia. Here's another primary spermatocyte, and these primary spermatocytes are going to be precursors for our spermatids. When we look at the primary spermatocytes, I want to emphasize that they have a relatively homogeneous purple nuclei. And then when we look at the spermatids, let's pull this up right here. The spermatids are right here, and they have a heterogeneous purple nuclei. So the nuclei of the spermatid is starting to enter meiosis, and because the chromosomes have condensed from the chromatin and are entering into meiosis 1, we can see some differences in coloration. The nuclei is heterogeneous. Now let's go back to our lab objective sheet here. I want to emphasize that we typically don't see secondary spermatocytes. That's within meiosis 2, and they just don't spend a lot of time in meiosis 2. It's just a split second. So finding those secondary spermatocytes on histology is just not something we are not going to ask you to do. And then we have the spermatids. These are going to be differentiated by early and late stages. As we are looking at the spermatids, the early spermatids are going to have much smaller, rounder nuclei, and they're going to be near the lumen of our seminiferous tubule. So there's an early spermatid there. Here's another early spermatid. I want to emphasize small purple nuclei near the lumen. And the nuclei themselves are also going to be rounded in appearance. And let's see if I can pull up some late spermatids. They're a little bit more difficult to see. I typically like going down here, or zooming out, barring my internet connection. The late spermatids are going to have an elongated nuclei. Small, dark purple, elongated nuclei. So I'm just scanning my field of view right here, trying to find a late spermatid, and I am not seeing that late spermatid readily available. So I'm going to punt down here and go and look at our histology of the seminiferous tubules. As we look at those seminiferous tubules, we can see in the lumen of the seminiferous tubules are late spermatids much more clearly. Assuming we find one. You know, class, with the difficulty I'm having with finding a late spermatid with our digital histology resources, I'm going to go ahead and make the unilateral call here and remove that from the lab objective sheet. Just because if I can't find it on histology guide and I know exactly what I'm looking for, it's not reasonable for me to expect you to find it. And then finally, we have the spermatozoon. This is just your fully mature sperm. Spermatozoa is the plural term. Oftentimes, we'll look at a different view of the male reproductive system. Let's pull up normally there's a sperm cell. So I'm just going to go ahead and go to the search option and type in sperm. When in doubt, you can always type in the term you're looking for and histology into a image search engine, and you're going to get some great results. So here we go. Here we have some spermatozoa, also known as fully matured sperm. And I really like this view of the spermatozoa just because the spermatozoa are clearly free-flowing, floating within the lumen of the vessel that they're looked at, or more likely this is a smear from a semen sample. So that's all we have on the male reproductive system's spermatogenesis. Let's move on to oogenesis. As we're looking at oogenesis, I want to first focus on the fact that this is an ovary. So here we can see an ovary. The first, and I want to use as a disclaimer, the fact that when it comes to follicles of the ovary, there unfortunately is a lot of disagreement depending on what resource you look at. So stick with the list that I gave you. I tried to make this as objective as possible. So first, here's an ovary. Let's focus on the primordial follicles. Primordial follicles will be located typically near the outer edge of the ovarian cortex, and they're visible as these large oocytes surrounded by a simple squamous epithelium. So right here is, I'm putting some purple dots within the nuclei of those oocytes. And in particular, those are primary oocytes. And then because they're surrounded by a simple squamous epithelium, we refer to that as a primordial follicle. The next follicle I want to cover with us is the primary follicle. As we look at a primary follicle, it's going to be similar to the primordial follicle, except the key difference I want you to fixate on is the cells that surround it. Instead of being surrounded by simple squamous epithelium or simple squamous cells, it's going to be surrounded by simple cuboidal cells or epithelium. So here is a primary follicle. That one appears to have a stratified cuboidal epithelium, but I want to highlight here, this is a good one. To be really objective, I want to emphasize simple cuboidal epithelium surrounding that primary oocyte. So we're still looking at a primary oocyte. As we look at this view right here, this may be confusing for students because there is not the purple nuclei present in the middle of that oocyte. The reason you can't see the purple nuclei is because the oocyte is so large that there are times where you can slice the oocyte and not see the nucleus in the center. Slice it in such a way that you remove the nucleus. Next we have the secondary follicle. As we look at secondary follicles, those we define as having 2 or more layers of cuboidal cells, so a stratified cuboidal epithelium. And another key thing I want to emphasize about the secondary follicles is the appearance. Oops, wrong button here. Let's zoom out. Sometimes with our secondary follicles, we can just start to see a clear space forming in them. But the key thing I want to emphasize for you with the secondary follicles is they have 2 or more, secondary, think 2 or more layers of cuboidal cells wrapping around the outer edge of that follicle. And then finally we have tertiary follicles and mature follicles. The tertiary, come on, quick keys, don't fail me now. I'm minimizing this window, I'm tired of it popping up. There we go. So as we look at the tertiary follicle is going to have an antrum present. And as we look at the antrum, I want to emphasize that the antrum is this clear space within the follicle, and this is going to be filled with follicular fluid. Now as we look at this particular antrum, I'm going to categorize this as a mature or Graafian follicle. And here's the objective way I want you to do this. Look at the longest cross-sectional distance or diameter of your oocyte, and then look at the cross-sectional distance of the antrum. So because our oocyte has a diameter less than the cross-sectional distance of the antrum, we're going to call this a mature or Graafian follicle. Let me clear the screen here. Here is a more stereotypical view of a mature follicle. Notice how, as we look here, that secondary oocyte, which is now going to be present at the mature follicle, is much, much smaller than the cross-sectional distance of the antrum. So the one that I had just highlighted for you on the screen, over here, this is more of an in-betweeny-weeny. I wouldn't use anything like this on your lab exam. On your lab exam, I'm going to focus on something that's really clear-cut for you. So, for example, this is very clearly a mature follicle with a very large antrum relative to the secondary oocyte. Let's see if I can find a good tertiary follicle for you. I'm not super pleased with this view. So let's pull up this overlay. This is a pretty dependable view for us here. So here we can see a nice mix of follicles. So here, I'll zoom in on this one for us. This is a classic mature follicle. The antrum cross-sectional distance, the short portion, is still much larger than the cross-sectional distance of that secondary oocyte. Over here, this is what I would call a classic tertiary follicle. So we can still see that there is an antrum right there, but that antrum is very underdeveloped. The cross-sectional distance of the secondary oocyte, or oocyte, excuse me, primary oocyte is much smaller. So those are our follicles. Let's talk about parts of follicles. We've talked about primordial through mature. Let's talk about oocytes. For your sanity, primordial through tertiary follicles, we are defining as having primary oocytes. Once that antrum is double or at least double the cross-sectional distance of the oocyte, we now have a mature follicle, and we now will have a secondary oocyte within that mature follicle. Now let's focus on some of the cells of oogenesis. We have granulosa cells. Those granulosa cells are going to be the cuboidal cells I had been emphasizing. So for instance, here we can see some cuboidal cells around the around the follicle. Those cuboidal cells are the granulosa cells. They can be either simple cuboidal or stratified cuboidal in appearance or presentation. So I'm zooming out a bit, just a skosh. Here we can see a stratified cuboidal epithelium, a very stratified cuboidal epithelium. So those are the granulosa cells. We look at those granulosa cells to help us differentiate between primary follicles and secondary follicles. If there's a single layer of granulosa cells or simple cuboidal epithelium, it's a primary follicle. If the granulosa cells are stratified, so we have a stratified cuboidal epithelium, we call it a secondary follicle. We also have some layers of the follicle I want to talk about. And to talk about those layers, I want to pull up here our follicle. So I'm not loving that one. Let's go with this guy. That's a pretty good one right here. Because this shows all those key layers pretty nicely. So we're still going to have those granulosa cells as the stratified cuboidal cells wrapping around the outer perimeter. But then we also are going to have some cells that are more squamous in appearance, more of a stratified squamous epithelium. Those are referred to as the theca cells. On our lab objective sheets, I'll pull it up for us. That's the theca folliculi. A lot of sources will say that there's an internal and external theca folliculi. We're just going to group them all together and say that all of these squamous cells that are superficial or exterior to the cuboidal cells will be the theca folliculi of the follicle. We also have around our secondary oocyte, so here's the nucleus of the secondary oocyte, here's the cytoplasm of our secondary oocyte. You can see there's this dark pink membrane. We refer to that dark pink membrane as the zona pellucida. Let me pull that up on our lab objective sheet. It's the zona pellucida. And then wrapping around superficial to the zona pellucida, technically the zona pellucida also includes this clear acellular space as well, but I really like to fixate on that pink membrane right here. And then superficial to the zona pellucida, we have a stratified cuboidal epithelium that's just around the oocyte, and it's within the antrum. This stratified cuboidal epithelium is referred to as the corona radiata. These are cells that radiate outward from the oocyte, and they go around the outer perimeter, kind of like the corona of the sun goes around the surface of the sun. The cells of the corona radiata go around the zona pellucida of our secondary oocyte. It's also worth emphasizing the zona pellucida is an acellular membrane or extracellular membrane. that's superficial to the secondary oocyte. So let's talk about the corpus luteum and corpus albicans, our remaining structures. As the secondary oocyte ovulates, the cells that are left over, all those theca folliculi cells and granulosa cells that are left over after ovulation, are going to turn into a structure known as the corpus luteum. Here on the screen, I want to highlight the corpus luteum for us. It usually is going to be darker stained or absorb more pigmentation during the staining process. In unstained specimens, typically has a yellow coloration, which is why we have the root word lute in it. This corpus luteum typically will appear around day 14 of our average menstrual cycle, or ovarian cycle, I should say, in this context. And then usually it will start to shrivel up and get smaller and smaller and smaller towards the end of the ovarian cycle, or monthly cycle, to be more generic. So, and as it gets smaller, it goes from this large, highly pigmented structure to a smaller white structure made of fibrous scar tissue that has a higher concentration of adipose in it. This structure right here that I'm circling is referred to as the corpus albicans. Albi is a root word that refers to albino or white. So the corpus albicans is the white scar on the surface of the ovary. And I believe that's all we have for stages of oogenesis. Let's talk about the menstrual cycle. I should have, if I was really planning ahead, I should have loaded all these images up in advance. Here we go. One of them loaded pretty quickly. You never know with my home internet. All right, they're all loaded. Fabulous. The stars have aligned. I can actually load images. So as we're looking at the menstrual cycle, the three phases of the menstrual cycle are the menstrual, proliferative, and secretory phase. Another name for menstrual cycle is uterine cycle. So while we're looking at the uterine cycle, let's start off with the menstrual phase. Menstrual phase is when the functional layer of the endometrium is the thinnest. So while we're looking at our endometrium right here, we can see there's necrotic fragments sloughing off. The functional layer of the endometrium is becoming much thinner and sloughing off. And the basal layer, shown here of the endometrium, is much thinner and will be left behind. This is typically day 1 through day 5 of your average uterine cycle. And after about day 5, mensing stops, so menstrual bleeding will cease, and then we go to proliferative phase. During the proliferative phase, the functional layer of the endometrium is becoming much thicker, and it's regrowing itself. So let's go ahead and zoom in on that functional layer of the endometrium. Maybe not quite that much. Here we go. So here we can see how it's been regrowing. And while it's regrowing, I want to point out the uterine glands. The uterine glands themselves are very linear in appearance. They aren't very convoluted, and they have very small lumens. That's another key characteristic I want to highlight for you as we look at the proliferative phase. So I'll zoom in super duper close on some uterine glands for us. They are relatively linear and have a very small opening or clear space or lumen on the inside. That was the proliferative phase. And then finally we have the secretory phase or secretory phase. This is going to occur after ovulation. So it begins day 14, and it's day 14 through typically day 27 of your average menstrual cycle, day 27 to 28 of your average menstrual cycle. So let's pull up the secretory phase. And as we're looking at the lining of the uterus, the endometrium, look at this functional layer. So the stratum functionalis. And as we zoom in on those uterine glands, holy moly, look at how convoluted they are. Now, instead of being relatively linear, come on, load, baby. You're working so well, Internet. We can see how they're convoluted and will go back and forth. I also want to emphasize how they have a very large lumen. The clear space within the uterine glands is much more pronounced within the secretory phase. And hey, it finally loaded. It only took, 30 seconds to load a picture. So that's all we have for these phases of the menstrual or uterine cycle. And that's everything we need to cover for discussion of gametogenesis and the female cycle. If you have any questions or comments, please feel free to post them in the class discussion board or shoot me an email. And as always, happy studies.